Hi guys, welcome back to our podcast. I'm Ava.
I'm Haiti.
And I'm Emma.
Today we're going to be talking about the CAR T cells, which is a type of therapy that is used for patients that have cancer to try and kill out all the bad cells. So today we're going to be talking about how it was invented, what it is, how it gets in the patient's body, some research methods, and then looking into the future, the pros and cons of it. But first, to understand what a CAR T cell is, you have to understand what a T cell is, because it's just a modified version of a T cell.
So a T cell is a type of lymphocyte that develops in bone marrow and helps protect the body from infection, for example, cancer. This happens from tracking proteins called antigens on the surface of the intruder and it attaches to major histocompatibility complexes. Each T cell is unique because it creates a design to fight for only one intruder.
And the major histocompatibility complexes is so important because the cell isn't able to differentiate what's an intruder or not, so it has to bind to this in order to be able to fight only one certain thing, or else that's when you get immuno issues, because then your body's just attacking everything.
So a CAR T cell, founded by Dr. Carl June, is a genetically engineered T lymphocyte where a chimeric antigen receptor, CAR, is expressed. CD8+ in CAR T is typically a cytotoxin lymphocyte that helps directly destroy tumors through mechanism of perforin and enzyme release. CD4+ in CAR T can also be a helper cell by supporting the function by releasing cytokines, which stimulate and coordinate immune response.
So then there's a bunch of different types of CAR T cells, and like we said before, they're each specific to a certain type of cancer. So they don't treat all types of cancers, but here's the list that they do treat. So now that you know what a CAR T cell is, how they get into the patient's body is by first having their own T cells taken out through leukapheresis, which is just out through their blood. And then in the lab, they are introduced to genes that code for CAR, often by using viral vector lentivirus. And then the intracellular interaction domain activates T cell when CAR binds. These CAR T cells are cultured in the lab until there are millions of them, and then they're reintroduced into the patient's body through an intravenous injection in their bloodstream. Once they are in the body, they will recognize and bind to their cancer cell-expressing targeted antigen, and then the binding activates and releases chemicals to destroy the cancer-specific cell.
Know all that, we're going to talk about the research methods of how Dr. June came about discovering novel antigens, which are new or newly discovered molecules that elicit an immune response for CARs. He did this in many different ways, including isoform peptides from RNA splicing for immunotherapy target screening. This helps to identify tumor-specific antigens by analyzing RNA splicing patterns, as well as long-read RNA sequencing, which allows for the discovery of mRNA splice junctions that can be screened against the tumor to identify these novel tumor-associated antigens. He also evaluated CAR T cell efficiency and specificity in many ways. This includes cold culture assays, which is when CAR T cells are combined with positive and negative control target cells. They line up to assess their ability to kill cancer cells. Also, cytokine analysis is when supernatant from cold culture is analyzed for secreted cytokines, which indicate cell activation.
So then he also did some research, and he used flow cytometry and high content imaging, which was used to analyze cell viability and other cell surface markers. He also used different models for preclinical studies. He used immunocompromised NSG mice. He also used syngeneic mouse models, which were immune competent mice with transplanted tumors that were used to study the interaction between CAR T cells and the host immune system. He also used in vitro models, which were cell cultured systems and organoids that were used to mimic tumor microenvironments and study CAR T cell behaviors. And then some other research areas that he spent some time was the engineering of cars, like improving their design and use of second generation cars. He also was looking at gene editing technologies like CRISPR-Cas9 that was used to modify T cells and enhance their potency.
Although these have helped many patients, some side effects include cytokine release syndrome, neurological problems, dyspnea, low blood pressure, aphasia, headache, seizures, tremors, fatigue, easy bleeding and bruising, hypokalemia, and hyponatremia.
So then here's like a video that we found of a patient that was able to benefit from the CAR T cells.
I call it my cancer marathon because it has just been a long, continuous cancer journey that has lasted over 22 years. My first cancer diagnosis came in the fall of 1994. I was 30 years old, a wife, a mom, working full-time. So I've had it all. Chemo, radiation, surgery, a bilateral mastectomy. I had to have my eye removed and a stem cell transplant. So I have had longer remissions that have gotten shorter and shorter. Ian, we are relatively newlyweds. I had my first relapse a year after we had started dating. Cancer has involved our entire relationship. So when I got this last diagnosis of lymphoma, it was clear that the standard of care was no longer an option for me. The navigator that was available to me to help me look through clinical trials said, would you consider a CAR T trial? It's so new that even my local oncologist last year, he wasn't aware. I honestly wrote to every single doctor that was leading a trial all over the United States. CAR T, when I first heard about it, sounded incredible, but yet so sci-fi, like this is how we're gonna treat cancer someday. And I was surprised that that someday was here.
It's your own body doing the healing, and they're only helping your cells. They're just giving them a homing signal to go after the cell they should be going after in the first place.
I entered the CAR T trial at Mass General Hospital in Boston. I received 3 days of chemotherapy, very low dose, and the purpose of that was to clear out the existing T cells that I had so that there would be room for my new supercharged, awesome T cells. 2 days later, I was admitted to the hospital to receive the dose. The first sign that the CAR T was working was actually on day 3. My side effects were pretty much the fever. I was a little bit achy. I think I had my blood pressure drop. And then I had severe pain in the area that the tumor was at. That was kind of exciting because the doctor said, that's the CAR T is going to work on your tumor. For me, immunotherapy was dramatically different than any other treatment that I'd had. I liked it because I felt like it was working with my body instead of against it.
And a few weeks after the treatment, we're walking Boston. We got the job done. Now let's go enjoy ourselves.
So my CAR T treatment was received last year. And then in October, I had a relapse, knowing of some of the things in my life that would be coming up, like my daughter getting married. You know, it was hard to think that maybe I wouldn't be here for those things. So I felt desperate, like I've got to get that other CAR T because I know it worked for me the first time. And when they did the PET scan, there was no lymphoma anywhere. They feel that somehow the CAR T got re-energized. I was back in remission. In my history of cancer treatments, I've had to be really persistent to stay in the game. Cancer Research Institute is doing the same thing. And I'm so appreciative of the effort to get this way of treatment available to people, because without that immunotherapy, I wouldn't be here today.
Now that we're coming up on a year, and I see how Jamie has responded, it's like we have a new life to start over.
And we finally took our honeymoon, now planning my daughter's wedding, and Now I can't stop thinking about grandchildren, and we just have so much to look forward to now, and I really think I'm going to be here for all of it. I'm looking forward to a 50th wedding anniversary. I mean, that would make me over 100, but I think we can do it.
I think we could do it.
We can do it. Yeah.
After viewing this video and learning about CAR T cells, here are some pros and cons going into the future. Some pros are that they are highly specific. They target cancer cells directly using engineered T cells with minimal impact on healthy tissue. There are long-term effects, which some patients experience lasting remission, especially with blood cancers. There are personalized medicines tailored to each patient's immune system, making it a powerful form of individualized therapy. And there are advances in solid tumors. Progress is being made in adapting CAR T-cell therapy for harder to treat cancers like brain, lung, and pancreatic tumors. Unfortunately, there are some cons though, like equity and accessibility. It is a very costly and complex therapy and can exceed $400,000 per year due to manufacturing, hospital care, and follow-up. There are some severe side effects like listed earlier, and there is also limited access since it is only available at specialized cancer centers, often not covered fully by insurance. This is not a universally effective treatment since it's more successful in blood cancers like leukemia and lymphoma than in solid tumors. Fortunately, there are some advancements though, like off-the-shelf CAR T cells. This is using donor T cells to make therapy faster, cheaper, and more accessible. There's also dual target CARs designed to attack cancer cells, expressing multiple markers to prevent relapse. There are safety switches, which are built-in switches that can deactivate CAR T cells in emergencies. And finally, there are solid tumor targeting, which there's more research into overcoming the tumor microenvironment.
So then there is also some concerns with the CAR T cells, like overactivation of the immune system, and this puts patients at risk of severe immune responses or damage to the healthy tissue. There's also like long-term unknowns because it is newly invented, that the effects over decades are still unclear since, yeah.
Yeah.
And then there's also access inequities. So like wealthier patients in countries are able to benefit more than if they were poor, and that increases healthcare disparity. And then manufacturing challenges is that they were that the personalized nature makes large-scale production difficult and inconsistent because it has to like be tailored for each specific person. So then in conclusion, there's going to be like more research done over the years, and continued innovation is going to make it safer, cheaper, and more versatile, but real challenges remain in terms of access, cost, and expanding effectiveness. But with careful research, regulation, and equity, CAR T-cell therapy could become more a mainstay in the fight against cancer, and like so more people could have access to it. So I hope you enjoyed our podcast and learned lots. Yeah.
Thanks for watching.
Thanks for watching. Bye, guys.